Sneha, shall we start the class?
Sure, Rani.
So, very good evening to all my dear students. So, today we have with us Dr.A.Snehalatha Rani Madam, Senior Scientist, Plant Pathology, to deliver a lecture on disease management in banana. So, I am very happy to introduce Dr.A.Snehalatha, my friend. She is working as a Senior Scientist, Plant Pathology at Dr.YSRHU, Horticultural Research Station, Kovvur, and her field of specialization is molecular plant pathology, integrated disease management, and biological control. Madam has joined as scientist plant pathology at Citrus Research Station, Tirupati in the year 2009. Later, she got transferred to HRS Ambajipeta in the year 2010 and worked for 6 years on coconut. Later, she got transferred to Horticultural Research Station, Kovvur in 2016. And presently, she is working as Senior Scientist Plant Pathology, looking after ACRIP, AICRP on fruits pathology projects at HRS Kovvur. And she has developed protection technologies for management of Sigatoka leaf spot disease of banana under high disease pressures and non-chemical management of post-harvest diseases of banana, carried out diversity analysis of Ganoderma isolates of coconut through pathogenic virulence studies, isogyme analysis, and PCR-based studies, optimized the dosage and frequency of application of Trichoderma resi and neem cake mixture for the management of Ganoderma wilt disease in coconut. She has successfully demonstrated the above technology in 270 acres of coconut fields in East Godavari and Srikakulam districts and supported by Coconut Development Board. Then she has also carried out DNA fingerprinting of released varieties from Dr.YSRHU and molecular characterization of biocontrol agents Trichoderma resi and Trichoderma hargianum. Carried out 6 projects funded by Coconut Development Board as principal investigator and co-PA and successfully completed the projects. She has also received Best Presentation Award from Dr. N.K. Krishnakumar, Honorable Deputy Director General, during 25th Annual Group Meeting on ACRIP on Palms at Kasargod held during the year 2016. And as team member, she has received Chaudhary Devi Lal Outstanding ACRIP Award, ACRIP Center Award for the year 2014-15. And as a team member, she has also received late Amit Singh Memorial Foundation Award for Best ACRIP Center on Palms for the year 2011-12, awarded at Madurai, TNIU during 11th Annual Group Meeting on ACRIP on Palms from 11th to 13th July 2012. And to her credit, she has published 20 international research papers, 13 national level research papers, 2 review articles, 4 book chapters, 10 technical bulletins, and 12 popular articles, as well as other publications. So with this brief introduction, now I would like to invite Dr.A.Snehalatha Rani to please deliver a lecture on disease management in banana to the participants of Banana Graduate Readiness Program. Thank you, Sneha. Please carry on.
Thank you, Rani, for the nice words. Is the screen visible on my screen now? Hello?
Sneha, it is visible. It is
Okay,
clear.
okay. Thank you. Good evening, dear students. Today we will see the disease management in banana. In this lecture, I am going to cover the importance of banana pathogens, and then what are the various types of banana diseases, particularly to Andhra Pradesh, and then what are the characteristic symptoms of these diseases and their mode of spread, long distance or short distance, whatever it is, the mode of spread of the pathogens, and finally the management of these banana pathogens. What are the different management measures and the biological or chemical or whatever the integrated management measures. And before going to the various types of diseases in banana, I would like to share the importance of this banana pathology. You might have learned in your fundamentals of plant pathology subject that plant diseases plays a major role by causing epidemics which lead to entire wiping of crop or crop varieties. This banana is one of the such examples where certain varieties are ruled out because of this disease epidemics. For example, this Panama wilt or Pizarian wilt caused by the fungal pathogen Pizarian. It is a major devastating disease from 19th century itself, and then still it is posing a major threat to banana cultivation all over the world as well as in our state. And during the 1950s, the banana is majorly dominated by one variety that is grown shape. And this variety belongs to the AAA genome, and having the export the value, this variety got completely almost wiped out by this Panama disease caused by Pizarian wilt by the end of 1960s. Nearly 40,000 hectares of this Crow-Michelle variety, it was replaced with the other AAA genome, that is Cavendish group, this subgroup Cavendish group, because of this Panama wilt. This is a situation where the disease posed a change of crop variety in a particular crop. And this is a picture showing the devastating nature of this Panama wilt in banana in Netherlands. And this is the cut open pseudostem portion of banana, where you can see the pinkish discoloration visualizing the Pizarian Oxysporum species Codens pathogen in banana. And another such example you might have learned in your subject that the disease triangle, where for an epidemic to occur, there should be three important factors, that is susceptible host, virulent pathogen, as well as favorable environmental conditions coupled with time, the fourth factor. And here that Sigatoka leaf spot is one such example where you can see this all the key factors such as susceptible host granule and virulent pathogen, that is Sigatoka, are right now it is being termed as universal leaf spot in India. And then under favorable conditions such as rainy season, you can see almost all the granule cultivation. If it is at the shooting stage, there will be devastating losses in the crop. Another such example is varietal resistance or susceptibility, beautifully depicted in this picture, where you can see this is a 6-soil horticulture resistation Kovvur. You might be learning that some varieties are resistant and some varieties are susceptible for a particular pathogen, whether it is fungal or bacterial. They are susceptible to the pathogen. Here you can see when we screened around 120 banana germplasm under sick plot conditions. The soil is sick for Pizarium. So the screen studies are sick plot conditions of Pizarium. Here you can see every 10th row, the Amrutpani variety, which is very susceptible, it was planted after every 10 banana accessions. You can clearly see all the 4 Amrutpani plants within 3 months, 2 to 3 months, we can see the susceptible nature of the susceptible variety to Pizarian wilt. And here is one such example, and I say that pathogens are more choosy. And one such example, other such examples are Amritsani is highly susceptible to Panama wilt. Telechakrikali is highly susceptible to rhizome bacterial disease, whereas granule is highly susceptible to Sigatoka spot or Unice spot. And another important we can discuss is production of quality planting material. What is the necessity here? The availability and use of tissue culture plants in banana, it made a very useful impact on crop growers. The crop is being infected with 4 major viral diseases, that is banana black mosaic virus, banana bunchy tuck virus, cucumber mosaic virus, as well as banana streak virus. So to prevent the spread of all these 4 viruses, the tissue culture technology is very useful in banana cultivation, and availability of quality planting material helps in prevention of these major dreadful diseases. And the last but not the least, the banana pathology necessitates the importance of integrated disease management. Knowing the field, whether it is infected with a particular pathogen or not, we have to select a suitable resistant variety. We have to select a quality planting material along with clean cultivation, and we have to follow the cultural management, including biological control, chemical control, and even such as rhizome weaving or an nematode management should also be taken care while imposing the integrated disease management. Then only we can control the banana diseases. And still today, this Panama wilt is a major problem. Even when including all these integrated disease management measures, we are very difficult to control the disease. And coming to the various diseases, banana is infected by all the bacterial, fungal, as well as viral diseases. And in each group, there are majority having major impact on banana cultivation. Coming to the fungal diseases, there are 2 major diseases, that is Sigatoka as well as Panama wilt. And here we will see the Sigatoka disease. Sigatoka, as the name indicates, the yellow. There are 3 types of Sigatoka diseases: black Sigatoka, yellow Sigatoka, and then Unice leaf spot. This the yellow Sigatoka, earlier this was the first recorded Sigatoka disease in Java and later in Sigatoka Valley of disease. Hence, it is called as Sigatoka disease. The pathogen is Mycosporella. And the species may be 3 different species: Mycosporella physiensis, Mycosporella musicola, and Mycosporella elusu. And the pathogen classification is, it is an Ascomystis fungi belonging to the class Bacteriomystis and subclass Bacteriomycetidae, and belonging to the order Capnodiales and family Mycosporellaceae. This is the sexual form of the fungus, that is Mycosporella. And here it is Mycosporella elusu, whereas the anamark is called as Pseudocercospora elusu. Coming to the symptomatology, we can clearly see under favorable conditions in a susceptible variety, these are the various symptoms that you can see. That is light green narrow specks of about 1 mm length on the upper leaf surface of the banana. It is the initial symptom, which later turns to streaks of more than 1 mm length and width, which are parallel to the veins. This is the characteristic feature of the Sigatoka leaf spot. The symptoms are always parallel to the veins. This condition we have to take into consideration, whereas other leaf spots or micronutrient deficiencies, they won't be multiple spots and they won't be parallel to the veins. Later, these streaks elongate and then expands laterally to become elliptical and turns rusty red. After elongation, these they become expands laterally, and then the tissue quickly turns brown and center of the spot later shrinks, and even the sunken area becomes gray, and surrounded by halo darker region. These mature spots are normally 4 to 12 mm in length. This is the symptom development from light green narrow speck to mature spots with a gray center and then sunken area. Under severe conditions, these spots coalesce with one another, and you can see on the entire leaf surface, you can see blighted area. And even after complete drying of the leaf, you can see the elliptical or spots, sunken spots of the areas where the disease occurs. You can see clearly on this brownish area also. And under susceptible conditions, if the disease occurs after the shooting of the crop, you can see the plant here. Usually in banana, after shooting, there won't be leaf production, fresh leaf production. And so, whenever the disease, if the rainy season coincides after shooting period, and if the severity of the disease is more, we can see all the leaves are infected with this disease, and this is the severity of the infection. Only 1 or 2 leaves that are erect, and then they are also dry. And because of this condition, even though the shooting emerges, the bunch emerges, they are half filled. And because of there is no green leaf area availability, and because of there is no photosynthesis, you can see unmatured banana, they are ripened, yellowing or ripening of the not properly filled banana fruits. This is the effect of this severity of this universal leaf spot under susceptible or favorable environmental conditions such as rainy season. And coming to the disease, earlier it is in India also, earlier it is called as Sigatoka leaf spot. Actually, it is a complex disease. One is black Sigatoka caused by Mycosporella physiensis, and Pseudocercospora physiensis is the NMR. Whereas yellow Sigatoka, it is caused by Mycosporella musicola, Pseudocercospora musae is the NMR. And then recently, this universal leaf spot. This is actually a sectorial leaf spot, but the sexual form is Mycosporella species only. And now recently, it is confirmed as Mycosporella enusae, the third species in Sigatoka complex, and the anamorph is Pseudocercospora enusae. And according to the NRCB, this is the enusae leaf spot is the only subspecies that is available in India. However, many papers speculate that all the 3 species of Mycosporella, they may co-inhabit the plant, and there may be chances of coexistence of these pathogens, which needs to be studied. And these are the photographs of initially split, black Sigatoka and yellow Sigatoka, and these are the comedian dimensions. Through visual pictures, it is very difficult to identify whether it is universally spot. To certain extent, universally spot we can differentiate because the size, the lateral elongation of the spots at later stages, it will be more than compared to the yellow Sigatoka and black Sigatoka. And these are the photographs and comedian structures. Even from comedian structures also, it is very difficult. Only molecular identification can differentiate the 3 subspecies, 3 species of Mycosporella. The pathogen, it produces mycelium of the castor sporodochium inside the palisade tissue of the leaf. And at the time of Comedia 4 and Comedia emergence, these are this tuft of sporodochium, it produces bottle-shaped Comedia 4, which erupts from the epidermal cells. And on these bottle-shaped Comedia 4s, you can see the production of Comedia, long cell, 5 to 6 celled, elliptical, cylindrical cells that are Comedia of universal leaf spot. And the pathogen spreads through this comedia as well as ascospores. Both sexual and asexual life cycles occur in banana plant. This is a healthy banana plant. Either an ascospore or comedium. It infects the spore after falling into the leaf tissue. It germinates and penetrates the leaf tissue through stomata, and inside the leaf tissue it develops and produces lesions on the leaf, and then after severity of the infection increases, it produces both conidia as well as ascospores. In conidial production, we can see the sporodokia and then this Uroseptospora like conidia, which again continues the secondary cycle and then produces multiple number of conidia, causing severe devastation. And in sexual cycle, it produces spermagonia and spermatia in leaf, and then it is a heterocalic fungi, and after fertilization, it produces perithecia with acyl and ascospores, and these ascospores, two-celled ascospores, they are used for long distance transfer. They travel through, they release it through rain splashes, forcibly release it from the leaf surface and mix in the air currents, and then leads to long distance dispersion of the pathogen, thereby continuing the life cycle. And coming to the disease cycle in epidemiology, these conidial spores, this is the asexual form of multiplication pathogen. They are produced in the presence of dew or rainwater. They germinate. There should be a small layer of dew or rainwater for the conidiospores for the production as well as for the germination. These conidia are discharged by rainwater and spread to the other leaves or other plants. Whereas ascospores, they are dispersed through wind for the long distance spread of the disease. And these ascospores germinate within 2 to 3 hours of deposition, provided a thin film of water should be there. And the optimum temperature for germination is 25 to 29 degrees centigrade. For ascospores, it is little bit less, 25 to 26 degrees. Whereas for the conidia, it is up to 25 to 29 degrees centigrade is the optimum temperature. And here you can see the incubation period for susceptible varieties is varieties, it takes 105 days. For the spore, after the landing on the leaf, after germination, and by the time of symptom production, it takes 11 days in case of susceptible variety, very fast when compared to the resistant varieties, that is 105 days. That's why in the susceptible variety granule, the symptoms are more prominent and more severe, and within 1 to 2 months, you can see entire defoliation of the leaves. And another important factor you have to study in Sigatoka spot is the disease. Here you can measure not the person disease incidence, because whenever the Sigatoka disease appears, it is an aerial borne ascomycetes fungi and spores may be there everywhere, and all the plants will be showing 1 or 2 spots. So 100% incidence will be there. So here, in case of polio diseases, we measure the disease by using person disease index, that is disease severity. For a particular disease, may be having a different disease scale, and based on the disease scale, we calculate the person disease index. And another important factor that we have to consider in case of unicellular spot or Sigatoka spot is youngest leaf spotted, because this is the this youngest leaf spotted is nothing but the age of the youngest leaf with 10 or more spots that correlates well with disease intensity. That is, in case of granule, a susceptible variety, the youngest leaf spotted will be 3rd or 4th leaf. However, in resistant varieties, it may be sealed. So in case of disease, Sigatoka leaf spot disease, this youngest leaf spotted in case of susceptible variety is very less, like 2nd or 3rd leaf itself, they got infected with these unicellular leaf spots. So there is severe intensity of this disease on yield in case of granule variety. Coming to the epidemiology, it is observed that relative humidity at both the hours, maximum temperature and minimum temperature. These are the important weather factors which influence the progress of Sigatoka leaf spot. And shooting to harvest is the critical time for the crop. Because if favorable weather prevails at this time, the loss would be greater. And in susceptible variety, the best time to plant is May to August in our state conditions. If you want to go for a compulsorily a granular variety, and if you want to avoid this Sigatoka disease severity, you have to plant the plants during May to August. And coming to the integrated disease management, compulsorily you have to follow the integrated measure. One is the recommended species should be followed, because the farmers in case of Alzheimer's they go for spacing 1.5 to 1.5 meters, and then they harvest the plants, because many number of they could accumulate around 1400 to 15 plants per an acre instead of 1200 plants, and they may be getting 300 to 400 more number of branches from the extra plants. However, in case of rainy season, and if Sigatoka disease appears at the time, because of the closer spacing, there may be changes in the microclimate, there may be increases in the relative microclimatic conditions, which will lead to favorable conditions for the pathogens. If that conditions prevails, it is very difficult to control, even though if you apply the chemical fertilizers, the chemical pesticides. And second important factor is the disease is prone to appear in rainy season, so proper drainage facilities to be taken care of under those rainy conditions. And then compulsorily you have to apply the recommended dose of nitrogen and potassium fertilizers, because you know that potassium fertilizers, they provide resistance to the pathogens. So the farmer has to apply the fertilizers, and then desuckering at sanitation and weed management, because the pathogen may survive on particular weeds. Complete sanitation of the field and removal of the weeds may remove inoculum level from the field, and thereby whatever the other management measures we follow, they will be effective. And another important factor is removal of the dried and infected leaves. This is very, very important, because on these infected and the dried leaves, there will be the ascospores and cornelia. The life cycle is being continued in those as infected and dried leaves. From these infected and dried leaves, if the dried leaf is 30% or more than 50%, then compulsorily that leaf has to be removed before spraying any chemical. That is another important cultural measure that to be taken care of in case of integrated disease management of Sigatoka. And another coming to the chemical measures, there will be prophylactic sprays as well as curative management measures. Prophylactic management is nothing but these two chemicals, spraying of mancozeb or chlorothalonil, and these chemicals, they are prophylactic in nature. They should be there on the leaf surface by the time of spore germination. Then only they will be able to control the spore. After germination, even though if you spray mancozeb or chlorothalonil, it may not be effective. Similarly, for coming to the curative management, these are systemic in nature, and they may be effective even after appearance of the disease. And one such fungicide is propiconazole. Spraying of propiconazole 1 ml per liter of water along with 10 ml of mineral oil 3 times at 25 days interval. It will take care the Sigatoka disease under low disease conditions. However, if there are continuous rains, if there are the rainy season is up to 2 to 3 months, it was distributed to up to 2 to 3 months, there are continuous rains, you have to follow a vigorous procedure. Here it is up to 7 sprays you can follow. The spray schedule is first spray as 0.1% of propiconazole And the second spray is, it's a combination product, carbendazim plus mancozeb, that is 0.1% of this combination product, followed by third spray of 0.14% of combination product, that is trifloxystrobin and tebuconazole, and the fourth spray is 0.1% of difenoconazole. These are the 4 sprays, and if needed, if severity is not yet come down, then you have to repeat again these about 3 sprays. So total 5 to 7 sprays depending on the severity, depending on the weather conditions, we can follow 5 to 7 sprays of these sprays. But they should be stopped before 45 days of harvesting. Otherwise, in the fruit, you may observe the pesticide residues, and which may be detrimental for export. And here, finally, you have to remember these 4 important points of, in case of Sigatoka disease management. That is, removal and burning of disease leaves is a must before chemical application, proper spacing to be followed, and prophylactic measures, they have to be taken care before rainy season. And then whatever this fungicide spray or any micronutrient spray, in case of banana, always you have to use sticking agent. It is compulsory because of the leaf texture of the banana. And these are some other not other. This is septoria, is nothing but universal leaf spot. Here you can see this is the symptom of septoria leaf spot. Earlier it was regarded as a septoria leaf spot. The sexual stage is mycospirilla only. Asexual stage, they call it as septoria. Now it is being called as mycospirilla unice. And this is another leaf spot, cardinal leaf spot, caused by cardinal unice. And here this is the symptom pattern. Large, pale brown, oval to fusiform. These are the shapes of the lesions, which become necrotic later, and a center often with light gray center. And you can also observe the concentric rings. When compared to the unice or Sigatoka, these spots are larger in size, and then not parallel to the veins. Sometimes they may be parallel, but not always. They are parallel to the secondary veins. And then we can also see concentric rings surrounded by a dark brown border, and yellow halo is visible in case of cardinal leaf spot. But this is a minor leaf spot. Appearance is observed, but it is not that much severe as universal leaf spot. And whatever the management measures we follow for universal leaf spot, they are also applicable for these minor leaf spots. And another important fungal disease in case of banana is Fusarium wilt or Panama wilt, caused by Fusarium oxysporum formospecies cubans. And this is also an Ascomycetes fungi belonging to the class Sardariomycetes and the order Hypocreae, family Nectriaceae, genus Fusarium, and Oxysporum formospecies cubans. And coming to the symptomatology, the disease, the pathogen is a soil-borne pathogen, and it produces both external and internal symptoms in the plant. When the disease appears on the susceptible variety or any variety of the banana, on the visual symptoms, you can see as external symptoms. That is, one is yellowing of the oldest leaves. In addition to the yellowing of the oldest leaves, which slowly spreads to the above, middle leaf and the upper leaves, and then longitudinal of the pseudostem, as well as petiole buckling. The petiole leaf, the leaf breaks at the leaf base and hangs around the pseudostem. That is petiole buckling. And another external symptom may be reduced leaf lamina and membrane and death of the plant. If you go to a field and if you observe these symptoms, you may attribute it to Panama wilt. In addition to that, some characteristic internal symptoms you can observe in banana are, in case of rhizome, if you cut open the rhizome horizontally, you can see reddish to dark brown discoloration of vascular tissue, which is characteristic symptom. And the tissue is very hard, even though there is reddish or dark brown discoloration. If you press, the tissue is very hard, and you can also observe blackening of feeder roots. These are the photographs of external symptoms. In the first photograph, you can see the splitting of the pseudostem. And the second photograph, you can see the yellowing and petiole buckling. At the base of the leaf sheath, you can see breaking of the leaf and then hanging around the pseudostem. And then third photograph, it is the leaf lamina, number of leaf as well as leaf lamina. It got reduced, and then this is the symptom of one of the symptoms of fusarium wilt. And in the last picture, that is complete death of the plant. These are the typical characteristic symptoms of fusarium wilt. This is fusarium wilt race 1 in Amrutapani. I'll tell later what are the different races. And then coming to the internal symptoms, this is the vascular tissue in rhizome. This vascular bundle, we can see complete circle of the reddish to black color of the vascular bundle. This is the characteristic symptom. And then even if you press also, the tissue is very hard, because why I am saying is, in case of rhizome rot also, you can see the same similar symptoms. Some of the symptoms are similar, not all. But then internal tissue, it is very soft in case of rhizome rot, but here it is very hard. This point you have to remember. And in case of fusarium, there are 4 major races. One race, race 1 that attacks Amrutapani. Our seniors, our near people, they use it to eat this Amrutapani variety. However, we may not see Amrutapani cultivation now in Andhra Pradesh because of this race 1. However, this Grow-Michelle in other countries and all, they got removed because of this race 1. And this race 1, it infects the cultivars of AAA group, Grow-Michelle, and then POM and its subgroups, then Spink. Our Amrutapani comes under this silk group, AVB, and then AVB group, Pisangavad. These are the 4 groups of banana that race 1 can attack, and it completely removed our Amrutapani because of this race 1. And the race 2, it attacks blue go, that is a culinary variety, APB culinary varieties and its close relatives. They are being attacked by this race 2. And race 3, it won't attack banana, it attacks Heliconia species, flower species, and shows weak symptom in case of Grow-Michelle variety. And then race 4, this is very important, because this race 4, it attacks AAA group. Whatever the race 1 attacks, this race 4 also attacks. In addition to the dwarf Cavendish group also it attacks. So, because of Panama guilt in case of in 1950s and 60s, they changed it from Grow-Michelle to Cavendish group. The banana cultivation is changed to Cavendish group. Now, because of this race 4, the entire Cavendish group also is under threat. That is why you might have seen some WhatsApp messages that banana is being removed or extincted because of that only. But however, scientists are conducting trials, doing research to control this race 4 also. There are some successful stories. So we may not worry, but we have to take care. We have to we have to have the idea, and then we have to take care. So this race 4 is very detrimental. It affects the races of both the hosts of race 1 and 2, as well as dwarf Cavendish group also. And throughout the world, it is being reported, and in countries like Taiwan, Malaysia, Indonesia, China, Philippines, Australia, Oman, Jordan, Mozambique, Lebanon, Pakistan, Laos, Vietnam, and India. In India also, during 2016, it was reported by NRCB, National Research Center for Banana, at Bihar and Uttar Pradesh. In Katihar and Purnia districts of Bihar, it was first detected that race 4. And later, the NRCB reported the appearance of these disease in Uttar Pradesh Ghaziabad district and Madhya Pradesh Berhampur district and Gujarat Surat district. So this is a threat right now. There is a threat for banana cultivation. We have to take care. It should not be spread from these states to our state. So we have to make all our banana farmers to be aware of appearance of this tropical racer or TR4 and its impact on banana cultivation. And race4 also, if you observe the same type of symptoms, only thing is the host is here Grand Line, Cavendish group. Whereas in case of race 1, that is Amrutapani or Martaman, Rastali group, silk group, that is race 1. Here it is Grand Line. All the symptoms are same, like yellow, buckling, stem splitting. When you cut open the pseudostem, you can observe the pinkish discoloration on the pseudostem. And then if you cut open the rhizome, you can see the concentric black ring in the vascular tissue region. And this is the Fusarium oxysporum fungus under electron microscope. And coming to the morphology of the pathogen, it produces abundant and sparse aerial mycelium, and this may be white to pinkish color or a dark brown color also sometimes it may appear. And it propagates through micro and macroconidia. And yes, picture, this is the microconidia, 1 or 2 celled microconidia which are they are 4 to 8 cell, sickle shaped, very characteristic to Fusarium. You can see if you observe on the PDA plate, the first pathogen to appear, the first saprophyte or pathogen to appear is Fusarium. And it also reproduces the chlamydospores, which are very, very difficult. Like if your field is infected with the chlamydospores, then those maybe produce terminally or intercanthally in the fungus, in the mycelium of the fungus. But if in the soil, if it is infected with this chlamydospores, even after 30 to 40 years, they will survive. The pathogen will survive in the field. So because of that only, these varieties are being extincted. And the dissemination is through, it is a soil-borne pathogen. So through soil, and then through infected rhizome, sucker material, if the mother plant is infected, from the mother plant, the sucker plants will be infected. And in the fresh soil, again, if you take these infected rhizomes and plant it there, again, that soil will be infected with the disease. And then running water, in the running water, the micro or macroconidia or chlamydospores, they will be dispersed. And then farm implements and machinery, these are one of the important dissemination tools. Even our footwear, the farmers, if they go into the field with footwear and if the soil is somewhat muddy, like wet conditions, if it sticks into the footwear or clothes, then also this pathogen moves from with that soil to other fields. This for long distance transport of this pathogen, this footwear and farm implements and animals are important factors. And studies for so this is, we know that this is very difficult to control. Once establishes in the field, it is very difficult to remove the pathogen. So screening studies, identifying resistant varieties is important. So the susceptible accessions that are recorded at HRS Kovvur are Maypoovan, Silk, Sangavak and Bluebird. These are very susceptible to the race 1 of that Fusarium. Whereas resistant accessions are fire hybrids, Engambi KM5, Pisancillium, Williams and GCTCV 119 and 215. These are the resistant accessions that are identified under cycloid conditions of HRS Kovvur. And these are the overall susceptible varieties that are reported by NRCB across the country. Under Cavendish group, that is Basrai, Robusta, Harichel, Grand Line, Jehaji, Srimanti, like that. Under Rastali group, similar to Amrutapani varieties, Amrutapani, Martaman, Malberg, Patkapura, Nanjangad, Rasabanu, Nepuvan, Pachanadam. These are the susceptible varieties. And Pome group also Sirimalai, Virupakshi, and Montan and Bluko, and Proven group Manoranjitam, Pisangmas, Brahmichel, and Telchikirkeli. These are the some of the overall susceptible varieties in India. And what are the resistant varieties to DRESS4 in case of throughout the global level are? These are the Pisan Jerry, the AA group, and Cultivar Rose AA group, and Fire hybrids, GCP CV hybrids and Fire hybrids. These are the resistant varieties. And then some plantain accessions, and then Transonic Granite Banana. These are some of the resistant varieties we do have across the global level for managing tropical rice-4. And coming to the management, this is one of the dreadful disease of banana and very difficult to control. So all the integrated measures we have to take it. If it is not there in the farmer field, care should be taken that it should not be brought to your field. And so movement of suckers along with soil should be avoided. If you don't know whether it is there and are not in that borrower field. And compulsorily use tissue culture plant so that the infected material won't come through planting material. And then not only banana, under banana, if you grow elephant putium, and if that field is infected with a fusarium, if you move elephant putium also, the fusarium will come to your field. So movement of any plant material or any soil material should not be there. And then after, if you are still using sucker material, you should ensure that there is no disease from the wherever you are bringing the sucker materials. And then after bringing, the suckers should be treated with 0.2% carbendazim solution for 30 minutes. And if at all, if you observe 1 or 2 plants infected, go for drenching with 0.1% carbendazim solution from 2nd month after planting to 7th month after planting. And you also have to take a banana pest and nematodes also, because sometimes the nematodes they act as predisposing factors, whereas the pests, while movement, they carry this fusarium from one area to another area. So these pests and nematodes also they have to be taken care of. And coming to the bacterial diseases that affect banana, the major bacterial disease in our Andhra Pradesh conditions is rhizome rot. It is caused by Pectobacterium keratovorum. Earlier, it is caused as Erwinia keratovora. In case of this rhizome rot, we call it as Erwinia keratovora, and both Erwinia chrysanthema is also reported. And it is an Enterobacteriaceae, Pectobacteriaceae family belonging to the family Pectobacteriaceae, and Pectobacterium keratovorum is the organism name. The disease is being called as bacterial head rot or hot rot or tip over or pseudostem rot based on the symptoms. As the plant tip overs, it is called as tip over, and pseudostem got rot because of that it is called as pseudostem rot, head rot, and hot rot. And in case of this bacterial disease, the symptoms depends on age of the plant and severity of the infection. Nothing but if the field is infected, it is also a soil-borne bacterium. If the field is already infected, if you plant a newly planted rhizomes, they fail to come out. If they are infected, they are failed to come out. And the young plants, they show stunting and yellowing. And external symptoms on mature plants are weakened rhizome and sometimes rotting of the whole pseudostem, and which leads to toppling of the plant with a slight vindicales. If you see internally, brown or yellow water soaked regions in cortex and rotting of the culm, and characteristic bad odor specific to soft rot that will be observed. These are the different symptoms at various stages. In small plants, you see the scented leaf. It turns necrotic and it fails to come out, and the plant dies. And in case of a little bit older plants, you can see the stem splitting, and then yellowing of the lower leaves, and then fresh leaves are very small and show micronutrient deficiency like symptoms. And this is the characteristic symptoms. You can see in Amritapani also the symptoms. The Panama wilt also some of the symptoms. Buckling is not there in case of rhizome rot. However, yellowing and pseudostem splitting will be there. However, this splitted area is very soft and bad odor comes out from this area in case of rhizome rot. And because the pseudostem portion got infected and because of the rotness in the pseudostem region, the entire tissue becomes softens and then the plant topples down. That is why it is called as tip-over disease. And if you observe, if you carefully observe the differences between Xerium wilt and bacterial soft rot, here the internal vascular bundle becomes necrotic, even then, even after the plant completely dries, it stands in the field. It won't topple down, and center vascular tissue turns ring-like, black ring-like. But in case of rhizome rot, you won't observe ring. However, the pith region it becomes soft, and then pseudostem also soft color characteristic odor will come. And both wilt and rhizome rot also appear at the same time. This is the situation where the vascular tissue becomes blackening and then the center portion becomes, pith region becomes infected with the rhizome rot. Dissemination of this rhizome rot is through infected plant material, soil, as well as irrigation water. So whenever in the infected soil, if you go for Thalassaccharicale or susceptible variety, you have to observe and you should not go for blanket irrigation. Compulsorily, drip irrigation has to be followed. And coming to the management measures, this granite, Thalassaccharicale and Kovvur, these are the susceptible varieties. So if your field is infected with rhizome rot, don't go for these three varieties, go for other banana varieties. And do not plant these where if you are going after 4 to 5 years cultivation, again you are going to these varieties, it should not be planted between February to June because higher temperatures they follow rhizome rot pathogen. And then sufficient irrigation in summer months need to be taken care of, and use of disease-free planting material. In any pathogenic conditions, you have to go for disease-free planting material. And sucker treatment here also, COC 3 gram per liter of water, it has to be followed. And improve the soil health using biological agents. Pseudomonas fluorescens is one such important biological control agent. It improves the root activity as well as it controls the this rhizome rot Erwinia bacterial. So improve the soil health with these trichoderma and pseudomonas. And the infected plants you have to apply bleaching powder 25 gram per liter of water per plant. And always use removal and destruction of infected and dead plants from the field, which removes major inoculum level. And drip irrigation need to be followed. And if the field is highly infected, crop rotation should be followed with rice and sugarcane up to at least 8 years. And last, viral diseases. There are 4 major viral diseases. One is one important is banana bract mosaic virus, BBRMV we say. It is first reported from Philippines, India and Sri Lanka. Here you can see the mosaic virus is positive strand, single strand RNA virus. And in the plant, you can see the symptoms like mosaic patterns, stripes, and spindle-shaped streaks on pseudostem, midrib, or bracts. On any of these 3 parts, you can see apple-like symptoms, so streak-like symptoms, whether it is pinkish in color or in the second photograph, if you see clearly, some yellowish streaks are there. So either the spindle-shaped yellowish or pinkish spots on the pseudostem or on the bracts or on the midrib. These are the characteristic symptoms of BBRMV. And spread is any viral disease. The spread is through either planting material or insect pests. Here it is banana aphid, Pentalemon nigronervosa, as well as Apis coccipi. These 2 are vectors for this virus. And the virus also moves through infected plant material. And the virus is a non-angular flexuous filamentous ssRNA. belongs to the genus Potyvirus and family Potyviridae. It codes around 10 protein molecules, and this is the genome map representing the BVR-MV. And the other viral disease is BBTV. This is a single-stranded DNA virus, first reported from Australia, and it is distributed in South and Southeast Asia and African countries. These are the typical symptoms. Here it is, you can see few dark green streaks on the second or third leaf, infected leaf, first infected leaf, you see dark green streaks or dots on the lower portion of the lamina. And the following leaf shows whitish streak, and the successive leaf lamina becomes smaller, both in length and width. So it is called as choked appearance or rosette appearance. These are the symptoms of banana bunchy top virus. And then chlorotic upturned margins you can also see on the leaf. The leaves become harsh and brittle, and this gives the plant rosette or bunchy top appearance. Spreading through infected plant material. And then through banana aphid, Pantellonia nigronervosa. And this is a virus belonging to the genus Babovirus and then other nanovirus family. And it is transmitted in a persistent, circulating, as well as non-propagative manner by the banana aphid. And it can BBTV, it has at least 6 DNA components of 1.1 kb, and the one component coding for replication initiation protein, coat protein, movement protein, cell cycle link protein, nuclear shuttle protein, and one protein is unknown. It's still not yet known the function of that protein. So the BBTV genome is 6 DNA component. And another viral disease is banana mosaic. We call it as a CMV, cucumber mosaic virus. This is a single-strand RNA virus. And the disease is called as infectious sclerosis, heart rot, virus sheath rot, cucumber mosaic, or banana mosaic. Based on the symptoms, you can see these are symptoms, various symptoms of CMV in banana. And both severe strains and mild strains, they produce symptoms. Mild strains, they produce only diffuse mosaic or line pattern, such as these diffuse mosaic or line pattern that will be caused by the mild strains. Whereas severe strains produce necrosis of emerging figure leaves. In small plants, severe strains, they cause necrosis, and the center leaf, it won't come out, and the death of the plant will occur. Whereas mild strains, they produce only mosaic or line patterns, and then sometimes they can be deformed, curling of the leaves. It also is also observed. Here you can see curling of the leaves, and then deformation of the leaves, line pattern, etc. And in some cases, in the last picture, you see necrosis. So some severe strains, they produce necrosis symptoms. The center leaf becomes necrotic, and then it won't come out, and the death of the plant. In case of younger plants, you can observe because of CMV. And studies through infected plant material and banana aphid viscosity. And the virus belongs to cucumber virus bromovirus family, and it is a spherical virus containing ssRNA. And most isolates are 3 genomic and 1 subgenomic RNA species, encoding 3 proteins: movement protein, coat protein, and other proteins. And the last virus disease is banana streak virus, BSV, a double-stranded DNA virus. It produces symptoms like this line, chlorotic lining patterns as well as necrotic patterns, and these are the symptoms. Depending on the isolated pathogen, the symptom expression vary, and chlorotic flecking to lethal necrosis. Chlorotic fleckings as well to lethal necrosis, the symptoms may vary, and the most common symptoms are narrow, discontinuous or continuous chlorotic yellow streaks, which run parallel to the veins from midrib to margin. And in case of severe strains, you see at the leaf base, necrotic streaking will also be observed. On the reverse side of the leaf, you can see chlorotic patches. And studies to infected plant material and mealybug species of mealybug are vectors for this BSV. And the BSV is integrated into the BB genome of banana. So you may observe episomal BSV or endogenous BSV. This is the line pattern is the endogenous BSV, that is the BSV integrated into the banana genome. Green is the banana genome and violet portion is the endogenous BSV. Whereas in case of episomal BSV, that is the virus particle introduced by the vector into the host, which is not yet integrated into the banana genome, that is episomal BSV, and this is the genomic organization of this episomal BSV. It contains a circular dsDNA and the virions are non-enveloped bacilliform virions. And whatever the viral disease, all the 4 viral diseases are of major importance in banana, and the banana viral diseases, they won't kill the plant, but yield reduction, quality and quantity of the yield will go down. And one important measure we have to take care is the detection of the viral disease at the earlier time. Either by means of DNA based or protein based molecular techniques are available, and these are different various techniques being implemented for the detection of viral diseases in banana. That is immunosorbent electron microscopy, ELISA, enzyme linked immunosorbent assay, dot immunoblot assay, DIBA, and PCR, and then RT-PCR, real-time PCR, and then reverse transcription, loop-mediated isothermal amplification. This is another technique. And then lateral flow immunoassay. These are the different techniques that are being used for detection of banana viral diseases across the country or all over the world. And coming to the management of these viral diseases, only thing is you have to always use disease-free planting material, preferably tissue culture plants, and removal and destruction of diseased plants, and vector management using whatever the mealybug or aphids, you have to spray methyl-limton 2 ml per liter of water, and always maintain clean cultivation with removal of the weeds. And while this occurring, if a plant is infected with virus, you should not cut that plant and then move on to the next healthy plant. If that is the case, the movement of virus particle from the infected plant to healthy plant may take place. So care should be taken while desuckering the plant. Desuckering is one of the important activity in banana at regular intervals of time. And the other minor diseases are stalk rot or cigar end rot. These are the symptoms. And if you take care of all the other pathogens in the field, right from planting to the harvesting, these pathogens may not appear. They may be under stress conditions. And spraying with carbon dioxide 1 gram per liter of water, and use of bunch cover or boot leaf covering the stalk, there may be some important factors to take care of these 2 diseases. And these are some post-harvest diseases. You can base of the hand, cut hand, you can see crown rot or anthracnose, typical Coltrotrichum anthracnose spots like sunken necrotic brown spots. And then this is a cigar end rot in granule. This is one of the symptoms, both pre-harvest and post-harvest also. Cigar end rot will occur before harvest as well as after harvest. So these are some of the post-harvest diseases. If you maintain a field free from other universally spot or other leaf spots and maintain clean cultivation, these pathogens will also be taken care. And after harvest, if you still feel that anthracnose or crown rot, these pathogens are there, you have to go for post-harvest dip in potassium aluminum sulfate alum 1%, followed by packing in low-density polythene bags, 150 grams polythene bags with ethylene absorbent. It will take care, it will prolong the green life as well as yellow life, shelf life of the banana fruits will be improved after this treatment. And then even disease incidence is very low in case of these treated hands. And whatever the diseases, this is the final last slide. We can say integrated disease management involving all these components: the selection of resistant variety, quality planting material, clean cultivation, cultural management, biological control, chemical control, along with other pest and nematode control. These should be taken care. And thank you for, I thank the organizers, Dean ma'am, as well as Ramanandan sir, for giving me this opportunity and your patience listening. Thank you. Thank you, one and all.
Thank you, madam.
Thank you, sir. If you have any doubts, or
Yes,
you can ask me directly or chat, message me.
oh. So I request any student, if you have any doubt, please post that doubt in the chat box. Madam, one question.
Yes, sir.
Is there any disease which is not completely curable?
Any disease which is not completely curable. I say all the diseases they have to take it, like we cannot completely remove a particular disease, a particular pathogen. There should be existence of both the host and pathogen, both the things to be there. Which is the upper hand? How we control? If we control properly, crop will take upper hand. Or if it is left over without clean cultivation and all, the pathogen may take over the upper hand. But still, complete removal of the pathogen is not possible.
Madam, one more question. How to control root knot nematodes? One question. Hello. Hello, madam.
Harsha, please unmute your mic. I think madam got disconnected.
Yes, madam. Madam disconnected. Questions sir explain chestarani cheppanaru madam.
Okay.
Conclude cheddam madam meeting.
Yes. Harsha.
Okay.
So students who have any doubt regarding today's class, you can post your questions in the WhatsApp group so that we will forward them to the concerned scientist.
Okay, thank you, madam. So, on behalf of organizing committee of horticulture, Venkatamanagudem, I would like to convey my sincere thanks to